Hello and welcome to our AP Biology classroom. We are excited to give you all a presentation on our gel electrophoresis breast cancer lab. As a team, we have been investigating whether our 18-year-old patient Hannah will develop the BRCA2 mutation. We have also been using the information provided by our lab background sheet to determine the ultimate family pedigree tree, the family genotypes, and how the BRCA2 mutation may affect the generations to come. In this presentation, we will explain how this lab connects to all 8 AP Biology units. The BRCA2 gene is composed of a specific array of adenine, thymine, cytosine, and guanine sequences. When mutated, the functions of the gene are no longer the same and may cause a malfunction in regulation, which can lead to the development of cancer. When a molecular compound is disrupted, it becomes denatured, which means it loses its initial function. Once denatured, the molecular strands of the BRCA2 gene are able to be observed through a gel electrophoresis procedure, which separates molecules based on its length. The BRCA2 gene is a protein that is involved with repairing damaged DNA. It uses homologous recombination to do so by repairing homologous strands to damaged areas in order to introduce working DNA. BRCA2 also works to control cell cycle by ensuring repairs are processed as the cycle progresses. BRCA2 works to prevent tumors by ensuring DNA is repaired and without fault. Mutated BRCA2 genes can lead to cancer development, which is why it is commonly referred to as a tumor suppressor gene when not mutated.
So Unit 3 was about cellular energetics. So how do cells use energy? Animal cells use it through cellular respiration, plant cells use it through photosynthesis, and in anaerobic conditions, fermentation takes place. So when fermentation takes place, that means that 4 is made and 2 is used. So in result, only 2 ATP is made compared to cellular respiration and photosynthesis that make around 16 times that. And cellular energetics, by definition, is how cells produce and utilize energy to carry out various biological processes. So it's not just how they produce it, but also how they use it. Gel electrophoresis is used to analyze expression of genes involved in energy production. So it just shows if a patient comes in with low energy, most likely there is a problem with a gene being expressed in the mitochondria. Unit 4 was about cell cycle and cell communication. So in regards to mitosis, the phases are prophase, metaphase, anaphase, telophase, and cytokinesis. In cytokinesis, that results in 2 identical daughter cells being made. Errors in cell signaling lead to out-of-control cellular growth. So if there is not a signal in the phase of cytokinesis that tells it to stop multiplying, it will result in it multiplying out of control. And cell signaling includes direct cell-to-cell contact, signaling molecules, intracellular signaling pathways. And gel electrophoresis can see expressions of genes involved in cell signaling and identify errors. So if there's an error in the production of a signal that allows the cell to stop production of the cell, then through electrophoresis, it can show which gene was hindered and pinpoint what's actually going on.
Unit 5, Heredity. In this unit, we learned about the process of meiosis and how the gametes of both the mother and the father fuse together to create a zygote. We also learned about heredity and how different alleles determine which phenotypes are expressed in certain individuals. In the lab, we learned that breast cancer links to chromosomes 13 and 17, which are autosomal chromosomes. Because of this, it is important that Hannah gets tested for the BRCA2 mutation, as it may be passed down to her future children. Thankfully, gel electrophoresis can detect if Hannah has accumulated the BRCA2 mutation. Unit 6, Gene Expression and Regulation. In this unit, we learn that genes undergo transcription and translation in order to create a specific protein. This protein will allow the gene to turn on and be expressed in a particular phenotype. This can range from eye color all the way to hair color. Although gene expression is very beautiful and unique to each individual, overexpression of mutated genes can be very dangerous. In the lab, we learned that the BRCA2 mutation causes uncontrolled cell division, thus resulting in breast cancer. Thankfully, gel electrophoresis can be used to detect these cancer-inducing genes.
So Unit 7 was about natural selection or evolution, which is organisms with traits better suited for their environment to survive / pass on their advantageous genes. So agents for evolutionary change are mutations, gene flow, genetic drift, non-random mating, and natural selection. And through gel electrophoresis, we can study genetic susceptibility or the inheritance of genetic mutations, and is often used with population genetics. So it is to examine allele frequencies or mutated genes to infer effects of natural selection. So this is also used with the BRCA gene to see in populations if it is genetic or if it was something outside and it is something environmental.
Unit 8, Ecology. In this unit, we learned about different species and how they interact with each other and the environment. Every individual has a specific niche and do their part to contribute to the well-being of their habitat. Every species in a specific population is necessary for the stability of their population's trophic pyramid. In the lab, we learn that cancer disrupts the body's system, or habitat, and creates several health effects for the affected individual. These effects damage the environment of the body and can unfortunately result in a lifetime of lingering issues. Essentially, breast cancer acts as a predator to the human body. Gel electrophoresis can recognize cancer's disturbance in the body and how it is damaging the individual's DNA. Bioethics. An alternate treatment to chemotherapy is hormonal therapy. In hormonal therapy, the patient receives several injections or oral medication to lower progesterone or estrogen levels. This type of therapy is most effective for individuals that have the early onset of breast cancer or are currently in stage 2 of breast cancer. Hormone therapy is a great way to slow down the gene expression of the BRCA1 or BRCA2 mutations. Although hormonal therapy is effective, it does come with several pros and cons. Some pros include hormonal therapy slows gene expression for the mutation, it is very effective for the earliest stages of breast cancer, and slows growth drastically. The cons consist of the therapy will have a little effect on the stage 3 breast cancer, may come with several emotional effects such as mood swings or anxiety, and you will have frequent injections or medication intake.
So I did my bioethics on informed decision making regarding CAM. CAM is complementary and alternative medicine or holistic medicine. And one perspective that a healthcare professional might go towards their patient is to inform accurately and not judgmentally. So to inform with support, with patient-centered care, and inform the risks and benefits of switching or incorporating CAM treatment. Switching is often rooted in cultural and spiritual beliefs. Another perspective that a healthcare professional might have is to inform with deterrence. Often, CAM can give a patient a sense of a placebo, and they might think the CAM is really doing more than it actually is. So CAM, there are a lot of studies that say that it is not very significant, that your health is really the same, and ultimately, the effects of if your cancer is being affected, the effects are from your cancer treatment and not CAM. So a lot of patients get a placebo effect from CAM and not really an actual support.
Who present with the BRCA variant, it does not entirely prevent the possibility of development. The various side effects of the procedure include infertility and premature menopause, outcomes that might outweigh a patient's desire to seek the procedure. Patients should be empowered by medical providers, not compelled, to decide whether or not this procedure is best for them by being informed of possible risks, benefits, and outcomes. It is a medical provider's responsibility to discuss the facts regarding various treatments rather than to use compelling evidence to drive a patient towards a decision that might not be in their best interest. Prophylaxis is an invasive procedure that works by removing the mammary gland that has yet to exhibit signs of cancer. This is done in an attempt to reduce the development of cancer in the organ, but it does not ensure the total protection against future development.